Let's get started then. So when we talk about the carbon atom, it a lot of times lays the foundation for a lot of molecules because it can make single bonds for them. It can make two double bonds. It can make a triple bond and a single bond. So it's quite flexible and quite diverse in the types of molecules it can make. And not only that, once molecules are attached to it, it takes a lot to really break them apart, right? So it's not like hydrogen, where hydrogen is kind of promiscuous. It wants to be everywhere. It even it forms attractions with other polar um, atoms, even when it's already attached to something covalently, right? So carbon itself, it provides a lot of stability to molecules, right? And so if we think about what that means, right, um, it has a lot of really flexibility and a lot of variability in the types of molecules it can make because it can bind with just about everything, right? Or make a bond with just about everything, right? So all life has a lot of carbon based on it, and then for the most part, the carbons are just really kind of the stability of a molecule, right? And with a lot of molecules that we extract energy from, that we can utilize to break down to create ATP and energy, um, the carbon just is acting to hold the really important hydrogens together, right? Because it's the hydrogens that the cells use in order to make energy or ATP. And if you haven't put that together just yet, that's okay. I mentioned it the very first day of class, but we're going to be talking about cellular respiration and photosynthesis in our second unit. Okay? An organic molecule, right? So the term organic is kind of been used for some really weird things, right? So a lot of times, if you eat organically, that means that you're eating foods that are not GMOs. They don't have any pesticides. They don't have any types of um, other other types of chemicals that are used on them, growth hormones or whatever, right? But organic to us in science, right? Not in marketing, but in science, organic means that the molecules contain carbon and hydrogen, right? That's what organic means. And everything that's alive is made up of organic molecules, right? And so both of these molecules that you're seeing on this particular image are both organic, right? Because they have carbon and hydrogen, right? And so, but if you look at carbon and the way it is kind of the center part or the centralized portion of the molecule, that's because of the types of bonds that it makes, and it makes these molecules very, very stable, right? And so we say that carbon kind of provides the structural stability, or it acts as the skeleton of a lot of molecules. Okay, and that's really important to understand, not only biologically, but also, a lot of you who, if you're going into the life sciences, right, you're going to have to take chemistry also. What you're going to find out is chemistry is also will tell you that carbon is really important because of the types of molecules it makes, especially once you get into organic chemistry and biochemistry, of which everybody who is a science major, not computer science, but anybody who's a science major, whether it be biology or um, chemistry or biochemistry or microbiology or whatever, you're going to have to take biochemistry. It's a requirement. Okay. So if we think about this for a minute, right? Organic molecules are molecules that are found associated with and derived from organisms, right? And they have to contain both carbon and hydrogen, right? And as long as they contain both those things, are considered organic, right? So if we look at the molecule um, that has two carbons attached to each other and then surrounded by hydrogens, then that's organic because it has carbon and hydrogen. But CO2, right? It's got carbon and then two oxygen atoms attached to it. Well, there is no hydrogen there, so that molecule is not organic, right? That's what we call inorganic carbon. And so, from an industrial, from a medical, from from a biological, from a microbiological perspective, inorganic chemistry can tell a lot about an environment, but it's not an organic molecule because it doesn't have both carbon and hydrogen. It just has carbon. Okay. So here you can see that I've got some other molecules. Here's some really cool molecules, right? I have fructose, I have ribulose, those are both sugars. I have proline and amino acid, right? I have testosterone, proline's an amino acid, right? Building block of proteins. I have testosterone, right? And so testosterone, you can see, um, it's really this really cool structure, but it's considered a steroid or a sterol, which falls into the lipid categories, right? And so that's a really important molecule to both males and females. We both make it, right? Um, we make it in different amounts, but it adds to some of the secondary sex characteristics of a male and even the female, right? And then you have heroin, and heroin is a whole different class itself, right? But look at the molecule itself, right? And it can have a lot of effect on the human body, physiologically, biochemically, and pharmacologically, right? So these are really, really important molecules. And the idea of organic is an important concept to grasp very quickly and to hold on forever because you're going to be dealing, if you're in science, you're going to be dealing with organic molecules forever. Okay? So if we think about organic molecules, biomolecules means that molecules that come from life, and macromolecules are molecules that are macro, they're large, right? And so sometimes these terms, biomolecules and macromolecules, are used interchangeably, right? So if I asked you, if I asked you right now, could you tell me what the four macromolecules or the four biomolecules, let's just stick with macromolecules. Can you tell me what the four macromolecules are? Um, in biology or in biochemistry, can you tell me what the four macromolecules, can anybody tell me what those are? Ms. Campbell, you want to give it a shot? Carbohydrates, lipids, nucleic acids, and proteins. That is absolutely correct, right? And those things are so important. Those things are so important. And we're going to be talking about those, right? So we're talking about carbon and how it acts to provide stability or act as a skeleton of a lot of molecules. And then later, we're going to be talking about how these carbons come together to form these macromolecules that are essential for biological life, right? And so it's really important that we understand how those things are put together. Okay? Any questions? So if we delve in a little bit more about the biomolecules, right? So biomolecules are essentially organic macromolecules, right? The macromolecules, Campbell just told us, are the carbohydrates, the lipids, the proteins, and the nucleic acids, right? And so if we look at these structures, look how beautiful these particular molecules are, right? This is several glucose molecules in a chain, right? And so depending on how those glucose molecules are in a chain, do they branch? Are they alternating? Are they similar, but they're attached only by the first carbon of one glucose molecule to the fourth carbon of a second? We call that a beta 1,4 linkage. Well, if we look at those things, then that particular chain, long chains of glucose could be things like starch, they could be chitin, they could be glycogen, or they could be cellulose, right? Depending on how they are arranged, right? But those things have very different functions in biological living things, and we're going to talk about those, right? So here's a triglyceride level, and I always tell my students that it's so easy to recognize a triglyceride, right? Because it has a glycerol right here, right? And then it has three branches that make it look like an E, okay? And those, so you have a glycerol molecule here, and then you have those three branching things, and those three branching things are fatty acids, right? And so that's important to understand because if we eat too many carbohydrates and the body can't handle them all, right? Can't do anything. What the body does is it converts those carbohydrates to triglyceride, and then we store triglyceride and we become chunky, Okay. So if you wanna lose weight, if you wanna lose your levels of triglycerides in your blood, what you want to do is you want to cut down on carbohydrates, and then also you want to increase your output exercise. Right? You need both of those. You can't just give up carbs though, because carbs are so important. But but you can cut down on the carbs that are not very good for us, right? So instead of eating a donut or 4 donuts, then maybe you could eat an apple or a nectarine, right? And those are different. Or a cucumber, right? Those are loaded with carbohydrates, but they're carbohydrates that the body doesn't use as efficiently, and so therefore the body gets the nourishment it needs, but it's not having to store the excess carbohydrates, as if in the case where you would eat a bunch of donuts. Okay, and this is my this is my hardest time of year because we're going into the fall, and the fall pumpkin spice comes out, and everything is pumpkin spice, and that's my favorite, one of my favorite flavors. But there's a lot of carbs in anything that has pumpkin spice. Right, so you have to be careful. I'm not saying give up carbs. I'm just saying be careful. Okay. Yeah, me too, Jennifer. There's nothing wrong with loving carbs. Do it in moderation. Okay. Good. And then you can see the DNA molecule, and that's just sitting there, and you're like, well, there's not anything too unique to that, except if you start to look at the individual molecules that make up the DNA, right? Then it becomes very, very complicated. DNA is one of the most complicated um, biological molecules, macromolecules ever. It's the master molecule. It's what runs our entire body, right? It runs every single cell, and all those cells working together make tissues, and all those tissues come together make organs, and all those organs come together and make organ systems, and eventually you have an organism, and they're all thriving, or hopefully they are, because of our DNA. So all disease happens at the molecular level, right? So people think that, oh my God, he's got diabetes. That means he's got, a pancreas doesn't work. Well, you're targeting the actual organ, but it really happens at a molecular level. And so what happens is that certain types of cells and certain types of receptors on those cells are not allowing glucose to be taken up by the different cells of the body, and therefore there's too much sugar in the blood, which then causes a lot of problems. Everything from kidney failure to heart failure to blindness. It's just an amazing thing. Um, the way diabetes works, and people don't take it seriously, right? But it is a terrible disease to have. Okay? Any questions? Treatable, but not curable yet. Okay? So it's all about this carbon. And so remember, the valence of carbon is 4. That means it has to make 4 bonds right now. It can make 4 single bonds. It can make 2 double bonds. It can make a double bond and single bonds. It can make a triple bond and a single bond. It can do all kinds of great things, and therefore the variety of different molecules that can be made by attaching different atoms to the carbon atom or to chains of carbon, right, becomes really pretty cool to think about, because the variation and the diversity of molecules associated with carbon is almost limitless. Okay. So here again, you see the valence of carbon. You can see the first shell, the first orbital is full, but the second one has only got 4. That means that particular atom carbon has to make 4 bonds in order to fill its outer shell. And it does very readily. And so if you look at this, you can see, although the molecules are quite small, you can see the variety of um, of how carbon is being attached to itself, and then how other things are being attached to carbon. The blue lines are the blue patterns that you see here. are the longest chain of carbon, right? And so what a lot of people don't understand is that molecules are really three-dimensional. They bend and they flex and all kinds of really cool things, right? So you take it, look at it, and then you see where the longest series of carbons are, and then you name it based on how those carbons are in a line, right? So that's what that shaded area is talking about. I want to talk about that in just a minute. Okay. So if we look at these particular molecules, you see here there's four carbons attached to a bunch of hydrogen, and that's called butane, right? Here you have six carbons that are attached to a bunch of hydrogen, right? That's called cyclohexane. And then here you have three carbons, and there's a double bond between two of them, right? And so therefore that's called propene. And then you have a gas, right? And or it can be a liquid, but that's formaldehyde. And that particular structure right there, a carbon that has a carbonyl. This is a carbonyl. We'll talk about what that is in a minute, right? Carbonyl is a C double bonded oxygen bonded to a hydrogen, and that's called a carbonyl. In this case, it's called an aldehyde. If a carbonyl is in the middle of a molecule, like this, right? Does anybody know what it's called? Does anybody know what that molecule is called? It's called a ketone, right? And so ketones can be bad for us, right? It for us, for humans, if we have high levels of ketones in our blood, it's an indication that we're starving, that our cells are not getting enough nutrients. Right? Any questions so far? So if we ask about these four molecules, are these molecules drawn correct? Are they drawn correctly, right? And I know some of you are having trouble with this, right? So we're going to talk about this before we start lab today. But if we look at those, right, based on, again, our understanding of honk, that's right, Campbell, based on our understanding of honk and the valence, right? So the valence of hydrogen is 1, it can make 1 bond. The valence of oxygen is 6, it can make 2 bonds. The valence of nitrogen is 5, it can make 3 bonds. It has to make 3 bonds. And the valence of carbon is 4, it can make 4 bonds, right? And so Campbell's correct. All of those molecules are correct, right? There, each of them are correct. Okay. So another thing that is important when we talk about molecules is if we look at this, like cyclohexane, right? And we start to look at it a little closer, right? So this is kind of so when I put this into Blackboard, it makes it into a PDF, and sometimes it truncates things, right? So what you're looking for here is that there's supposed to be some more carbons attached to each other, right? So that it's showing essentially that there's 2 hydrogens to each of these, right? And when we draw it this way, that's a lot of stuff to write. So a lot of times we just kind of draw it like this. And this indicates this particular, this, um, this drawing right here, this ring structure here indicates this, okay? We just quickly draw it like this. Some of us, when we're drawing these things, or even go quicker, right? We'll just simply do this, right? And to us, that means it's the same thing. Okay, I just added a functional group to it. So please take a look at this presentation that's not in this lecture because you'll get to see the entire molecule instead of kind of just being truncated. Okay, so if we look at right here, you can see here's some other examples of organic molecules, right, that are all drawn correctly. And so here you can see this is benzene. And so if we look at cyclohexane versus benzene, what's the difference? What's the difference between cyclohexane and benzene? Can anybody see the difference?
The double.
It's got double bonds, right? It's got double bonds. And so when we draw benzene, right? You know how we drew cyclohexane and we drew it so that it looked like that, right? When we draw benzene, a lot of times we'll draw it, but we indicate those double bonds by just simply putting a circle inside of it, and that means that it's double bonded in there, right? There are alternating double bonds, okay? And that means that there's only one hydrogen attack. Because remember, carbon can only make 4 bonds. It has to make 4 bonds, right? So if there's a double bond alternating in the carbons in that particular molecule, then there can only be one hydrogen. Which is another thing I want to point out. Every time you see these structures, right? Every time you come to a point, right? Every time you come to this point, that indicates, let me draw it again. That indicates that there's a carbon attached at that point. Okay? Good? Any questions? Okay, so homework 2, day 2, asked you to draw, actually homework 4, asked you to draw some isomers. And there was some problems with this already for the ones that I looked at. And so there are different types of isomers. Isomers just mean that the molecule has the same types and the same numbers of atoms. Okay, so you can see that a structural isomer has the same types of atoms and the same numbers of atoms, right? So if this has 4 carbons, right? This one also has 4 carbons, right? But it also has how many? 1, 2, 3, 4, 5, 6, 7, 8, 9, 10. It has 10 hydrogen, right? Well, this other one, in order for it to be isomeric or be an isomer, it has to have 10 hydrogens. 1, 2, let me count them. 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, right? 1, 2, 3, 4, 5, 6, 7, 8, 9, 10. So this one also has 10 hydrogens, okay? And that makes them isomer. Oops, 10 hydrogens. That makes them isomers, right? A geometric isomer means that between 2 carbons, there's a double bond. And then on that double bond, either on one side, there's the same atom or the same molecule, or when it's like that, when you have the same, so here you can see there's a methyl group and a methyl group and a hydrogen, and they're on the same, right? You can see that, right? So that makes it what we call cis isomeric. But if we have it opposite sides, right? Hydrogen here, hydrogen there, and you have methyl group here, but they're on the opposite sides, right? And we call that trans, right? And so you all may have heard of trans fats. Trans fats are terrible fats for us. Don't eat them. A lot of times we make trans fats by frying food. So anytime you fry food, even if you fry them in olive oil, you're still producing trans fats, right? The worst fat that you can use, of course, is animal fat, right? Although it really makes things taste delicious. But those types of fats are not good for us. And you get them by increasing those fat levels to a high enough temperature that you make these trans fats, right? Okay. And then if they're enantiomers, right, that means that they're mirror images of each other, right? So if you were to put a mirror right here and you were to look at them, they're mirror images of each other. Okay? Everybody see that? Okay. Now, let me ask you a question. If I was to draw this particular molecule, And if I was to draw this particular molecule, I'm sorry, if they're little lines like this that don't have anything, it's basically understood that there's hydrogen attached to this. Okay? So here are these two molecules. Are they different? And if they're different, how are they different? Oh, Campbell says, no, they're not different. Campbell, enlighten us. Why are they not different?
One can be turned and superimposed on the other one.
That is correct, Amundo, right? So what I always tell students to do is look at the molecules critically, right? And so where that functional group is attached, that's where you count the first carbon. Here's the second one, and here's the third one, right? And here you have your functional group right here, right? That's a hydroxyl. So that's your first, that's your second, and that's your third. And like what Campbell said is if you just flip these over, they will fit right over each other. This is the same molecule, right? It's one propanol, okay? Same molecule. If we wanted to make it different, but make it into an isomer, there's only one way to do it, right? And that's to do what? Who can tell me? What do I need to do to make it different? Thank you, Miss Maria. And so you would put the hydroxyl on the second carbon. Okay, good. And so that makes this particular molecule 2-propanol. Okay, and even though the molecules are arranged a little differently, it still has the same number of atoms, right? And the same types of atoms, so it's isomeric. Okay, questions? And the reason I'm doing this is because people have been turning in homework and they're drawing this atom and this atom, or I'm sorry, they're drawing this molecule and this molecule, and they're the same. Okay, so the only way you can make them different is to put that hydroxyl, put that OH in the middle, and then they become different. Okay, any questions? All right, so again, if we look at cis versus trans, remember, and the different atoms or the different molecules that are attached to the carbon are being represented by the green structures, right? And so you can see on the top part, the green structures are on opposite sides, right? They're across from each other, and that's called trans. And if they're on the same side, they're adjacent to each other, and that's a cis isomer. Okay, those are geometric isomers. Okay, so those are real simple ones. They get real complex, right? So you can have these cis and trans fatty acids, right? And so, well, I will tell you that the trans fatty acids are really bad for us. We have to be careful that we're just not consuming too much fatty acids, right? We want to be sure that we are taking in really nutritious foods, right? And things that aren't going to be really bad and going to attach to our blood vessels and cause things like cardiovascular disease or cerebral vascular disease, things like that, right? We're going to be real careful of that. But you can see that the cis fatty acid has got a double bond right there, right? And then it's got the hydrogen on the same side, adjacent to each other. For the trans fatty acid, here's a double bond. Remember, these geometric isomers center around a double bond and 2 carbons, but you can see the hydrogen are on across from each other on opposite sides. Okay, good. Anybody have questions? Okay, So we now understand about isomers, and we understand about carbon and how important carbon is. And now we're going to look at functional groups. Functional groups are groups of atoms, right? Sometimes a molecule, that can be added to a carbon on another molecule, and it will completely change the way that molecule functions, right? And so there are several different types of functional groups, right? The ones that I'm going to ask you to know are hydroxyl, that's OH, right? Carbonyl, and so carbonyl, there are two types. They're at the end, if you have a carbon double bonded to an oxygen bonded to a hydrogen, that's called an aldehyde. But if the carbon double bonded to oxygen is in the middle, it's a ketone. Okay, so here's the aldehyde, and here's the ketone. Okay. You also have a carboxyl group, also known as a carboxylic acid. And here at the end of a molecule, you have a carbon double bonded to oxygen, which is then bonded to an OH or hydroxyl. That changes the structure completely and changes the way that that molecule functions, right? You could have an amino group or an amine that's attached to a molecule, and it changes the molecule. It makes it more basic, it makes it more polar, it does a whole bunch of different things, right? But amino acids have an amino group in it, right? And it also, and amino acids also have a carboxyl group in it, right? So the amino makes it the amino, and then the carboxyl makes it the acid. So you have these two things together, you have an amino acid, right? So you can see how adding these different functional groups to molecules will change the way that the molecules act. Okay. There's a methyl group, right? And if you add a methyl group, although there are lots of methyl groups that are on the molecule already, but let's say you have DNA, right? DNA is deoxyribonucleic acid, right? It has an acidic ending, but if you methylate that and you make that ending a methyl group, you methylate it, it becomes non-functional, right? And so that's the methyl. And then you have phosphate groups, and phosphate groups make things more acidic, they make things more polar, and they add to a lot of potential to molecules, right? And so you can add a phosphate group to the end of ADP adenosine diphosphate to make ATP adenosine triphosphate. And that molecule, adenosine triphosphate, will allow cells to do that. Okay? And then the last one is a sulfhydryl group, right? And so this sort of sulfhydryl group. Sometimes it's known as a thiol, right? A thiol. And they're slightly polar. Some people will say they're non-polar, but they're slightly polar. But what they do is they smell bad, right? So think of the way a skunk might smell, and those are thiols, right? And so I don't know if you've ever been sprayed by a skunk, but that smell is hard to get rid of, right? Sulfhydryl groups, there are other things that are important also, right? When they're attached in amino acids, they help to fold the amino acids, and so the folding of those amino acids become proteins. Okay, so I want you to know there are 6 of them, right? And here they are. Actually, there's 7 of them. Here they are, right? So I want you to know hydroxyl, methyl, carbonyl, carboxyl, amino, phosphate, and sulfhydryl. Okay, good. Any questions? So if I was to draw these things really quickly for you, right? A hydroxyl would look like this. A methyl group would look like this. A carbonyl at the end of a molecule would look like this. But in the molecule, it would look like this. Okay. A carboxyl would look like this. An amino would look like this. Okay. A phosphate group would look like this. and sulfhydryl group looks like that. And those are the functional groups. Okay? Any questions? Right, so here they are for you one more time. Right, hydroxyl, carbonyl, carboxyl, amino, sulfhydryl, phosphate. And the one that's not here is methyl, because methyl groups are found in a lot of different molecules, and in some cases they're already ending. It's only when we change things that are acidic or aldehydic that we change them to a methyl ending, and then they become very different. Right? They act very differently. Okay. I have a little bit of a table here for you guys to look at your own time. We're going to go over these now so that you can see how these things will change the molecule, but that's there for you all if you want to just do a little bit more research, right? So here you can see that we have some different types of hydroxyl groups being added, right? And when we add them to something, they really change the functionality of a molecule, right? So here, if we have an ethyl group and we add a hydroxyl group to it, it becomes ethanol, right? Or ethyl alcohol. A lot of people like that because, of course, that's the major alcohol that's in beer and wine and champagne and a bunch of other things, right? It's happy juice for some people. It's a depressant, right? So people don't know that. People don't know that when you take alcohol, it's a depressant. It depresses your mental state, but it also suppresses your immune response, right? Bad time to be drinking ethanol if you're in a large group with COVID out there, because it really stops your immune system from functioning properly, right? If we take 3 carbons and we put them in a chain, that's a propyl group, and we add a hydroxyl group to the middle one, we have now made isopropanol or isopropyl alcohol. That's rubbing alcohol. Now, rubbing alcohol is good for a couple of things. It's good to remove water from certain things. It's good as a real limited and very general anesthetic, right? But it's also poison to us. So we don't want to drink isopropanol. It's poison, right? And then glycerol, right? Which is found in a lot of fatty acids, but it's also found in a form like it in antifreeze, right? And glycerol can be really, really deadly to us, right? If we ingest it. If we put it into our body just a little bit, it's not problematic. And glycerol is used in vaccines because it helps to stabilize the proteins that are in vaccines and they don't fall out, right? There are 2 types of vaccines. The one that's got glycerol, if you take it, it stings a little bit when it goes in. The other one is an aqueous vaccine. It's made in water and it doesn't sting. Okay? Questions? So you can see just by adding these different functional groups to different things, different molecules that are already formed, you change not only the structure, but you change the functionality of that molecule, right? Case in point, here, of course, you have 2 men, right, in caricature, right? This male appears to be very fit, right? He's probably in his early to mid 30s, right? Testosterone is being produced quite a bit. Here's another guy, right? He's probably in his mid 30s to mid 40s, because as we get older, men start to produce less testosterone. And the testosterone is what makes us look good, right? And so here you can see that this other guy here, he's got a beer belly and he just doesn't have a lot of tone in his body, right? So if we look at testosterone, right, this is the way testosterone looks right here. But if we just change the functional group just a little bit, right, so you can see the difference right here, right, and right here. Then we make another steroid, which is estradiol, right? And so you can see that just by changing the functional groups, right, you can change the way the molecules react. Any questions so far? Okay. So functional groups, right? We want to look at these things and how they're going to change the molecule, right? And so let's delve into them a little bit. Let's look at them, right? First one we're going to talk about is hydroxyl, and that's what the hydroxyl looks like, right? When you add hydroxyls to things, it turns them into alcohols, or they can be alcohol sugars, right? So how many people have ever heard of mannitol or sorbitol? How many people have ever heard of those, right? How many people have ever heard of mannitol or sorbitol? If you chew bubble gum, right, the dentine stuff and all that, those are all um, sorbitol, right? So other sugar substitutes are mannitol. And our bodies do not handle those sugars very well. We don't break them down all that great. And so therefore they don't add a lot of calories to us, right? But also, you can make ethanol and isopropanol and butanol and pentanol. I mean, there's all kinds of different alcohols you can make. Almost all of them are deadly to us, except for ethanol. Even methanol is not good for us, right? But they can be used for a lot of different things, fuel sources, cleaner burning fuel sources, and so we like that. If we add a hydroxyl group to a molecule, it becomes more polar, right? It's water soluble because it's hydrophilic, right? And it's electrically neutral, right? So it doesn't really allow for a charge to travel through it all that easily. Okay, good. Questions? All right, let's look at carbonyls. So carbonyls, this is the hard one for people to get sometimes, right? Because it can either be in the middle of a molecule or it can be at the end of a molecule, right? If it's in the middle of a molecule, right, it's attached to carbons, right? So here you have a carbon here and a carbon there, and then you have the main carbon here that has a double bonded oxygen. That's a ketone, right? And so remember, in us, ketones are not good, right? And so a lot of times when they're doing biochemical testing on your blood or your urine, they're looking for increased levels of ketones. That says a couple of things. Number one, it might say that you have an injury to the body. It might also say that um, your kidneys aren't functioning properly, right? Because they're not cleaning out all the waste of the body. And the other thing it might say is that you're not eating well, right? Okay, ketones. Aldehydes are even more toxic, right? They're going to be things like formaldehyde, glutaraldehyde, those kind of things. Very, very dangerous molecules, right? A cleaner that is very popular, especially with the Hispanic community, is called Fabuloso. Anybody ever heard of Fabuloso? Who's heard of Fabuloso? Yeah. So if you've heard of Fabuloso, it is a tremendously beautiful cleaning agent. It works really great, but it has glutaraldehyde in it. So you should never ever use Fabuloso on anything that's going to come in contact with food because it's so toxic. Okay, you can use it to clean um, showers. You can use it to clean sinks. You can use it to clean toilets, but do not use it to clean plates or forks or spoons or anything like that because it um, it is very, very toxic. Okay, formaldehyde, for those of you who may know it, a very dilute form of formaldehyde is known as formalin, and it's what we use to preserve bodies when people have died, and we don't want their bodies to degrade very quickly, right? Because we want to be able to have a funeral service or a wake or something like that, and we don't want their bodies to decay too quickly, right? Because the body will begin to decay in about 24 hours, right? Um, and so we don't want that to happen. If the if the mortuary professional has done their job correctly, the the body's going to look like it's in what we call state. It's going to look like it's just inanimated. It's not moving. If the mortuary professional doesn't do their job correctly, the body will look kind of gray. That means they've added too much formalin, and so the body turns kind of gray. Okay. If we add a carbonyl group to a molecule, it becomes more polar, right? Um, and it's also electrically neutral. Okay. All right. Carboxyl. Carboxyls are found at the end of a molecule, right? So at the end of a molecule, here's a long chain molecule here, and then you have a carbon attached to this carbon, and then you have this double bond oxygen, and then carbon is also bonded to a hydroxyl. This is a carboxylic acid, right? This is what it's called, a carboxylic acid. And things that have these are going to be acidic. They're also going to taste sour. So all citrus is really has carboxyls in it, right? So lemons, oranges, anything that's of the citrus family, limes, any of those things, pomelos, any of those things are going to have carboxylic acid to it, right? They're polar. They make the molecules more polar, and they make the molecule acidic, right? So you can see that all acids have a carboxyl at the end of it. Okay. Any questions? All right. An amino. Amino can be added to any place on the molecule. Um, it makes the molecule more. It makes the molecule polar, and it makes it more basic, right? And so amino acids, but also if you think about it, chloramines. If you've ever heard of chloramine, how many people have heard chloramines? Anybody ever heard of chloramines? Chloramines are added to the municipal water system. It makes the chlorine a little bit more stable, and therefore it can act as a disinfectant longer in the water systems, right? So if the water treatment plant is, I don't know, I'm just gonna make it up, down on First Street, and you live 30 miles away, right? You want the chloramine levels to be high enough so that microorganisms and bacteria aren't going to grow in the water and make you sick, right? Be very happy that you live in a country like the United States that has the infrastructure so that most individuals that have a municipal water system have clean water coming into their homes that aren't going to be contaminated by some of the major microorganisms that can cause disease and infection. Okay? Questions? Okay. Phosphate groups, right? So phosphate groups can be added to membranes. They can be added to other molecules, right? And they make the molecule polar. They make the molecule acidic, and they make the molecule so that it acts as an anion. Okay. Any questions? Right. So examples of this would be phospholipids, right? It could also be things like ADP and ATP, and that's what's being drawn here, right? So here you have your ATP molecule. This particular part of a molecule, I like to call the HEB. Anybody work at HEB? Anybody work at HEB? Hey y'all, I like HEB. But here, the HEB stands for the high energy bond. And when that particular bond is broken, the molecule goes from being ATP, adenosine triphosphate, 1, 2, 3, to ATP, adenosine diphosphate. And this bond that breaks that last phosphate group releases energy. And now our cells can do work, right? And what we mean by work is they can break down chemicals like glucose, they can transport things across the membrane, they can do all kinds of different things, right? And we're going to be talking about some of the functions that the cell does in unit 2. Any questions? Okay. The last group, or the sulfhydryl group, and this is the way it looks, right? So you can add it usually at the end of a molecule, right? And so here you can see this is ethanethiol, right? And it smells bad, right? So it makes the molecule slightly polar. 8 years ago, I went to a conference and there was this great debate about thiols, about sulfhydryl groups, and whether or not they were um, polar or non-polar, right? And there was half the group in that conference, so about at this particular talk, there were about 80 of us. So 40 people in that group thought, you know, they're polar. The other 39 said they're non-polar. And I was kind of in the middle saying, I'm not sure, but I need you all to come to a conclusion so I can understand this better. They had this great debate. It was a lot of fun to listen to. But we believe it's slightly polar, right? It's hydrophobic. It doesn't like to mix. It doesn't like to mix with water all that much, right? And so therefore, if you get sprayed by a skunk, you're not going to be able to rinse off the smell with water. It's not going to be attracted to it. You're going to have to use something like baby oil. Okay? Good. Something to know in case you get sprayed by a skunk, right? And it makes the molecule more basic. Okay? One of the more important things that sulfhydryl groups do is they form these disulfide bridges, right? So if you think about this orange line as a long chain of amino acids, right? These disulfide bridges form, and what they do is they start to fold the protein, they start to fold the amino acids on themselves, right? And so what happens is as these proteins start to fold, you start to see kind of the shape of the protein be formed, right? And if you all remember that the functionality of a protein is established by its shape. So the shape of the protein is going to tell us what that protein does, right? This is the active binding site right here, right? And so in that particular case, a substrate would come in there, would attach to that particular binding site, right? And if it's an enzyme, then that substrate would attach to that binding site. And that enzyme could then break that apart into its two subcomponents, right? And if it is, for instance, the sugar, which is a disaccharide, it's got two sugars together, right? It would break it up to its two pieces, which would be glucose and the lactose. Okay? Questions? So the takeaway from this right now is with proteins, what establishes its function is its shape. Okay? Any questions? Anybody have any questions? You guys are very quiet today. Okay. So we're going to transition to talk about function. I'm sorry. We're going to transition from talking about functional groups to talking about macromolecules. On the macromolecules, we're going to talk about our carbohydrates and lipids and proteins and nucleic acids. And then after we finish nucleic acids, we're done with the material for the first unit of this course. And we're ready to take our first exam. So if I was to predict where that's going to end, right, I'll get through carbohydrates today, and then we'll stop, we'll take a break, we'll go to lab. Okay, so it looks then on next Monday, we will talk about lipids, and I'll start talking about proteins, maybe finish proteins. But next Wednesday we would finish Unit 1. That means that the homework, the exams will be about the following Monday. Okay. As we get closer there, I'll remind you also that you know. Okay. So we're going to start to transition from talking about functional groups to start to talk about macromolecules. Okay. So if we think about this for a minute, all of the macromolecules, with the exception of lipids, right? So carbohydrates, proteins, and nucleic acids. They have monomers and they have polymers. The only one that does not have monomers or polymers are the lipids. They have three different types, right? And we'll talk about them in a minute. But here you can see that the monomer is essentially one structure, right? Where a polymer is a chain of these structures all together, right? So now what was that? Don't know that yet. Don't know that yet. I'm sorry, I don't know that yet. Unless somebody's got an email that I haven't seen just yet. Haven't got an email about that yet. So as soon as I know, I'll let you know. Okay. I hope it is, but don't know. So if we look at the monomers, right, for carbohydrates, the monomers for carbohydrates are what we call monosaccharides. And there are 5 of them. And I'm going to have you know those things on the spot. If I catch you at HEB, I might quiz you. I might say, Campbell, list the 5 monosaccharides right now. Right? So I do that a lot to my students when I catch them at HEB or wherever. Okay. So the monomers of carbohydrates are the monosaccharides. The polymers are carbohydrates, and they have names, right? And we'll talk about that in a little bit. The monomers for proteins are amino acids, and of course, if you put a bunch of amino acids in a chain, you get a protein, right? Monomers are the nucleotides, and you put a bunch of nucleotides together, you get a nucleic acid. But for lipids, there are no monomers. There are just types of lipids. And the types of lipids are triglycerides, that's the first one. Second one are phospholipids. And the third are sterols, also known as steroids. Okay? So again, lipids, there are 3 types, but there are no monomers or polymers. Okay? Questions? So how these things are put together is what we're going to be talking about in a few minutes, right? And so if we think about these, they're put together by dehydration reactions. And what dehydration reactions simply mean is that water gets removed from the molecules. And when it gets removed from the molecules are put together. Okay? For instance, here we go. Right? So you can see here that you've got glucose and glucose, right? You take the water away, you take the hydroxyl away from this one. So you have hydroxyl from here and a H from there, you form water. You take that water away, but these 2 glucose molecules come together and they form the disaccharide, the two um, the 2 sugar molecule that's known as maltose, right? So where do you find maltose? Or what is maltose used for? Anybody know? Malaya? Where's maltose? And where is it? What does it do? Where is it? Where is it found? What is it? Anybody know where maltose is found? It's malt sugar, that is correct. So, Sonny, it can be used to make beer. It can be used in like candy, right? The malt, because it's glucose and glucose. Are you with me? Because it's glucose and glucose. So, um, if you put this candy, like a malt um, egg, like you find the candy at Easter, right? If you put it in your mouth, it's just gonna melt in your mouth, and they're good, right? But they're just glucose, right? Are you with me? So if you look at another one, right? If you have glucose and fructose together, and you remove water, you put those two together, and you get the two sugar molecule that we know as sucrose. What is sucrose? Table sugar. That's right. So most people think most people think that table sugar is glucose. No, I mean it does have glucose in it, but it's sucrose. Right? Are you with me? So dehydration reactions take water away, and then they put molecules together to form chains of molecules. Right? And so if we think about that, you can see that here is some real simple reactions that show that water being taken away leads to larger molecules. Okay? Any questions? The other type of reaction is called a hydrolysis reaction. And in this case, what you do is you add water, and the water really breaks things away. So here you can see you have sucrose right here. You add water, and you break the two the two sugar molecule apart to form its parts, right? So now you have glucose and fructose. Okay? So remember that. A dehydration reaction removes water from the molecules, but it binds the two molecules together, and therefore can make long chains of these molecules, of what we call polymers. Where a hydrolysis reaction will use water and break down reactions, right? And therefore it'll take these larger molecules together and make more simple molecules. Okay? Any questions? Okay. So let's talk about carbohydrates, right? Um, we're not going to get to lipids today. I've got about 26 minutes or so to talk about carbohydrates. I can do it, but we won't be able to finish the lipid portion today. So we'll finish the lipid part of this lecture on Monday, right? So carbohydrates, right? Really important macromolecule, right? And so they're found in lots of different things, right? They're found mostly in what you and I would call things that have carbs, right? Breads and pastas and fruits and vegetables and all kinds of different great things that we like to taste, right? And so again, if we remember, this is one of the macromolecules. And macromolecules are very big structures, right? They're biologically active, right? And so if we think about these things, then they're essential for life, right? Macro, large molecules, and they're biologically active. So everything that's alive, including microorganisms, bacteria, need to have these macromolecules in order to survive. Okay? So we're going to be talking about carbohydrates. We're going to be talking about lipids and proteins, and so and then nucleic acids. But today we're going to concentrate on the carbohydrates. So if you think about that, remember that a dehydration reaction removes water, and when it removes water, it combines these molecules together, making larger molecules, and eventually it can make chains of these molecules, right? And then hydrolysis reaction adds water, and when it adds water, it breaks up the molecules, right? That's important to understand because a lot of the enzymatic reactions that we'll see inside of a cell are going to be hydrolysis driven. And a lot of the biosynthesis that we see in cells are going to be dehydration driven. Okay? So if we think about carbohydrates, right, it starts with the monosaccharide. And so the monosaccharide we call simple sugars, right? And so the thing about what I want you all to know right away is that carbohydrates are composed of 3 atoms. What are the 3 atoms that they're composed of? Somebody tell me. Carbon, hydrogen, oxygen. That's correct, right? So on an exam-poo, if I ask you what macromolecule is composed of carbon, hydrogen, and oxygen, it's going to be the carbohydrate. Okay. So if we think about this, right? So the formula a lot of times for a lot of the carbohydrates is CnH2n and ON, right? And so if we think about that for just a minute, it's going to be, if you have 6 carbons, you're going to have 6 times 2, you're going to have 12 hydrogens, and then you're going to have 6 oxygens. Okay? And so that's kind of a real quick formula for figuring out the way sugars are put together. Okay? But really, they're easy to distinguish from each other because of the carbon, the hydrogen, and the oxygen are located. And there's different ways to draw them, right? We can draw them in a linear format like this. We can draw them in a linear format. Oops. Or we can draw them as a ring structure. Okay? Good. I am not going to expect that you're going to memorize what each of these carbohydrates look like structurally. But I will expect you to be able to tell me if it's a monosaccharide, a disaccharide, a polysaccharide, and I'll expect for you to tell me if it's a carbohydrate, right? Okay? But I'm not going to expect you to draw the individual sugar molecules. That's just, to me, that's not that important. Okay? Maybe if you go to biochemistry, it will be, but not for this course. All right, so if we look at these things, right, sometimes we can draw them much like we did with the benzene ring, right? We can take shortcuts. So here you can see that we are using kind of this little house to represent different types of sugars, right? If you see these things that have 1, 2, 3, 4, 5 ring or 5 carbons in a ring, that's going to be a sugar, right? Now there's another one here, but I'm only looking at the ring. Okay, good. So ribose and fructose, right? Those are going to be your, or at least those are 2 of your monosaccharides. I'll expect you to know every monosaccharide. You should be able to just spit them out. It's important, right? And so they're coming up if you don't know them already. How many people know? How many people know what the 5 monosaccharides are? Does anybody know already? Does anybody know already? They are glucose, fructose, galactose, ribose, and deoxyribose. Those are the 5 monosaccharides, and I want you to know those already. So if we think about these real simple sugars, these monosaccharides, they're the building blocks of the polysaccharides and the disaccharides, and we're going to talk about those also, right? So here's glucose. Glucose is needed by every living thing on the planet. Every living thing on the planet needs glucose. There is this process known as cellular respiration. It has 4 parts to it. It has this part that starts with glucose. It's called glycolysis, right? And then it has this 3 other parts, right? Which, if you don't know just yet, anybody know what the 4 parts of cellular respiration are already? Has anybody ever heard of that? Does anybody know what the 4 parts of cellular respiration are already? Sunny, do you know? What are they? Sunny, what are they? You can speak through your microphone if you want.
Man, it's been a while. Let's see. You got to go through the Krebs cycle.
Okay, but let's do them in order, right? So let's do them in order. So the Krebs cycle is the 3rd one. So let's just do them in order. So it's glycolysis. Then it's known The next part is known as the bridging step. Some people call it the prep step. The third part is also known as the citric acid cycle. Sonny calls it the Krebs cycle. That's good. And the last part is the electron transport system. So if you guys don't know that, you're in the right place. Right? Sonny, I'm going to bring you I'm going to give you a little bit of a refresher on it when we get there. Okay? One of my favorite things to talk about. I love to talk about cellular respiration. Okay? So if we look at this, right, you can see that here are some structural isomers, right? Glucose, galactose, and fructose. They have the same number of carbons, hydrogens, and oxygens. Right? That's why they're structural isomers. The five monosaccharides that I want you to know off the top of your head are glucose, fructose, galactose, ribose, and deoxyribose. You do not need to know what the structure looks like, but you need to know. I might have a question on the exam. Which of the following is not a monosaccharide? And I'll have glucose, fructose, deoxyribose, and galactose, and then I might have something like lactose, right? And the answer would be, which one of those is not a monosaccharide? Well, what's the answer? What's the answer? Lactose. Lactose is a disaccharide. Okay, so here are the 5 monosaccharides. The 3 disaccharides I want you to know are maltose, sucrose, and lactose, right? And so if you think about this, then maltose is made from, you should write this down, glucose and glucose. Okay. Sucrose is made from glucose and fructose. And then lactose is made from glucose and galactose. Right? Good. Any questions? You should know those. Gonna be on the exam. Okay. All right. And then we get to our polysaccharides. Our polysaccharides are those things that are in long chains. Right? Poly means many. Right? So some people refer to the disaccharides as polysaccharides because they have more than one sugar, but really they're 2, so they're disaccharides. But the polysaccharides, there are going to be 4 of them that I want you to know, right? On the 4 that I want you to know are cellulose, starch, glycogen, and chitin. Okay, so let's just make sure we know where these things come from. Where does cellulose come from? Plants. Very good. Plants. Do we as humans have the ability to break down cellulose? No, we do not. Therefore, if you eat any fruits or vegetables, then when you go to the bathroom, there they are, right? You swore you chewed up all the kernels of corn you ate, and there they are, right? You miss them, right? We do not have the ability to break down cellulose at all. Cattle do, right? Other ruminants do. Deer do. That sounds weird. Deer do. Deer are able to, right? But we as humans cannot. We can extract nutrients from them, but we cannot digest plants at all. Okay. Starch. Where's starch found? Potatoes and other plants, right? So potatoes, beets, carrots. There's a bunch of them out there, right? Tapioca root. Oh, there's a bunch of them, right? Delicious stuff, right? And these are where cellulose is supportive structures for plants. Starch is the way that plants store these long chain polymers, right? Glycogen. Who knows where glycogen is found? In the liver. That's right. That is exactly in animals, right? Yeah, it can be in other things. Bacteria have glycogen also, but fungi have glycogen also. But we as humans store our glycogen mostly in the liver, and then some of it, we have a little bit of it in our muscles, right? And it's really important because when you get up in the morning and haven't eaten breakfast yet, so you can go all day long, and if you're doing those fasting things, you might go, till 7 o'clock in the evening. But if you're not consuming any nutrition the whole day, then your body has used up all of its glycogen, and physiological changes happen to the body. Some people get shaky, right? They shake a little bit. Some people get loopy. What I mean by that is, what's 2 plus 2 plus 2 is 7, right? You get loopy, you're not sure, you forget things. But everybody gets really, really terrible breath. The breath becomes more metallic in taste, and it smells awful. You can always tell when somebody hasn't eaten, at least I can, when somebody hasn't eaten a meal that day because the breath is atrocious. Okay, good. So our bodies can take glycogen, can break it up into a whole bunch of glucose molecules, and then it can use glucose as a fuel to nourish every single cell in the body. Our body's quite remarkable. Okay? The last polysaccharide that I want you to know is chitin. And where do we find chitin? And arthropods, very shrimp and insects and fungi, very good. And prawns, very nice. You guys are using a bunch of cool words, right? Yeah. It's the exoskeleton of almost all the arthropods, insects, and arachnids, and crayfish, and lobster, and you can go on and on and on and on. And the fungi have it also, right? That's structure stability of the molds, and some of the macroscopic, like the mushrooms, right? And so there's a company here in town called, I don't know what it's called, but they are selling crickets, right? And they're using crickets as a source of carbohydrates. And they're mashing the crickets up. They're growing a bunch of crickets. They're mashing them up, and they're making a flour out of them, right? Because really, it's just a bunch of glucose. Are you with me? So I used to buy these things to show my different classes about them. So the marketing ploy for it is to call it an energy bar or to call it a protein bar. And they sell them like this, right? I don't know if you can see the little cricket guy on it, right? I guess you can't see it here. But you can see it says cricket. All right, can you all see that? It says cricket right there. Okay. So this is an energy bar, right? And they're pretty good. This one kind of has a flavoring of banana, but they also have cranberry and some other flavors, and they're pretty good. You can buy them. They were selling at HEB, but I don't think they were big sellers, so I can't find them at HEB anymore. But you can get them at Amazon. So they sell cricket chips, like potato chips, and all kinds of things like this. But if you think about it, folks, we as humans are taking up so much of the resources on our planet that we're going to have to figure out other things to eat, right? And so there's a lot of crickets, and there's a lot of roaches, and there's a lot of those things. So that they're all made out of chitin, and they all have a whole bunch of sugar in them, right? So we're going to have to learn how to eat those things, right? One of my best buddies in high school got to be in special forces. He was a ranger, and in order for him to pass a test in ranger school, if you will, they stuck him in the middle of the ocean, and they said, you have to swim 30 miles to this particular island, right? And at that island, you're going to spend a week, and you have to survive with whatever you can find, right? So he ate a bunch of insects, and he said that it didn't really bother him after that because every time we got together, he always kind of grossed people out by picking a spider up off a wall and eating the spider, right? But I don't recommend that for most people, but he did that quite a bit because really, they're just a whole bunch of nutrients if you think about them. Okay. Any questions? So, how many people? If we were in class, and so if we go back on Monday, I will bring in some of these cricket chips and these cricket bars, And everybody can try them. Mariel, are you willing to try it? No. Why not? Oh my God. Like to eat it, of course, Tamara. Why else? Why else would I bring them in just to look at them? Nah, we're gonna try them. All right, everybody's gonna get a try if you want to, Mariel. I'm not gonna make you try it, right? But I just think it's so Stephen. If you look at it, I they sell them as protein bars because that's a marketing thing. But if you look at this, if you look at this, the carbohydrates in it are 28 grams for this little bar that I just showed you. 28 grams of carbohydrates, but there's only 7 grams of protein. So is it really a protein bar, Stephen? What do you think? Is it really a protein bar?
So it's more like a pre-workout bar.
Yeah, exactly. It's exactly what that is, right? So it's carbohydrate. That's all it is. It's carbohydrate. But it's a marketing ploy, right? Because they call it a protein bar, right? Or an energy bar. But it's mostly carbohydrate, right? So 28 grams of sugar, it's like a little bit less than you would find in a can of soda, a can of Coke, right? And a can of Coke, I think it's 38. I haven't drank one in a long, long time, but I think it's 38, right? So that's a lot of sugar, right? So these are not bars that you want to eat all the time, right? A healthier version of that is the one that I eat on a tennis court. And the one I eat on a tennis court is called a balance bar. How many people have heard of those? That's what they look like, balance bars. Okay. They have a really good mixture of carbohydrates to fat to protein, right? So this one here, I'm pretty nerdy when it comes to nutrition, right? So the protein bar, the balance bar has got 200, right? And the other bar has got like 240. So it's a little bit more, right? But not substantial. Okay. Any questions? All right. So if we think about polysaccharides, some of them, like chitin and like, yeah, I don't like the protein drinks all that much either. They don't really have any taste to them. But I'll mix them in with ice cream. That's all I have, and I'll do that. That's not real healthy, but yeah, that's good. Okay. Anyway, some of the polysaccharides have these alpha or beta 1,4 linkages, or alpha or beta 1,6 linkages, right? And what that means is the first carbon and the fourth carbon are attached to each other, and that gives these things a lot of structural support. And I'll explain what that means in just a little bit. Okay? Questions? All right. So let's look at why carbohydrates, right? So the three types of carbohydrates, right, are of course monosaccharides and polysaccharides. And for us, they do a lot of great things, right? So some of them can store carbohydrates, like for instance, glycogen or starch. Some of them are structural carbohydrates, like for instance, cellulose or chitin. And some of them are helpful for cell-to-cell identification. So every one of our cells, Stephen, in our body, every one of the cells in Stephen's body, except the red blood cells, are labeled Stephen, right? So the immune system now knows which cells belong to Stephen because they are aware of the marking system, right? The marking system is based on this really beautiful mechanism that's genetic, right? I'm not going to explain it, but it's known as the major histocompatibility complex. If you don't know what that is, it's not part of this course, then don't worry about it. Okay? If you know what it is, oh, I remember that, right? But every single cell in our bodies are labeled us, right? So every single cell in Stephen's body is labeled Stephen. Every single cell in Logan's body is labeled Logan. Every single cell in Alberto's body is labeled Alberto, and every single cell in Jade's body is labeled Jade. Okay? So now the immune system knows, oh, that belongs to me, and I'm not going to hurt it. Or if something else comes in, yeah, if something else gets in, then um, then the body sees it and says, oh, it doesn't belong here, and they destroy it, right? Part of the immune response. Except the red blood cells. Red blood cells have their own marking system. They're A, B, AB, or they have no sugars on the surface, and they're called O, right? But now the body knows, right? So if Stephen and I were best buddies, and I was really hard on my liver, I drank too much, and then I started to have liver failure, and Stephen came up to me because he's a real big nutrition guy, and he kept me, you know, you really need to stop drinking. And I stopped drinking, and Stephen saw that I was really taking care of my body now, even though my kidneys were failing. And he said, well, you know what? Because I told him I'm not doing well. My kidneys, I'm going to go on dialysis. And so as a as a gift to me, he had his blood type checked out, and there was a good cross type and match between him and I. Not only were we good buddies, but he's decided he's going to give me the gift of life. He's going to give me one of his own kidneys, right? Hypothetical, not for real. But he's going to give me one of his own kidneys, right? Is my body going to accept, even though we're a good cross type and match, is my body going to accept or reject the kidney that Stephen has given me as a gift? What do y'all think? It's going to reject it every time. Why, Alberto? Why is my body going to reject the gift that Stephen has given me? Anybody? What do you mean by that, Sonny? You're using these fancy words, right? The body fights off cells that aren't. That's correct, right? And those are the immune system at play, right? So the immune system is going to recognize the kidney that Stephen has given me as not part of the body, and it's going to reject it. So, and even though we're good cross type and match, I would probably have to take immunosuppressants for the rest of my life um, in order for my body to be able to accept that particular kidney. And that's right. That's correct, Albert. It's a good way to put it. Good way to put it. Yeah. No, I can't be bribed. I'm sorry. That just can't happen. Right? Any questions? So cell-to-cell communication, cell-to-cell identification, we're going to be talking about that, right? In Unit 2, we're going to be looking at how these things work, right? And I think you're going to find it quite fascinating, the way that the body works and the way that our cells work. To me, it's just fascinating, right? Every day that I'm alive, and I get, I have a really cool job. I get paid to learn, and that's really cool, right? Yeah, I teach courses, but really, I get paid to learn because I do a lot of research, both application research, but also just book research, right? And I learn a whole bunch of things, and then I get to talk to you about them, which is pretty cool. Okay? Any questions? So, we talked about dehydration reactions, right? Yeah, you are really charitable, right? And we talked about the beta 1,6 linkage, right? So if we say beta 1,6 or beta 1,4, what we're looking at is how they're linked together. So let's look at beta 1,4, right? So the way we label these things, right, are by carbons. So here, this is the first carbon. Oh, I should use a different color. This is the first carbon, right here, that little peak, first one. Here's the second one. Here's the third one. There's the fourth one. And there's the fifth one. And then this guy right here is a sixth, okay? 6 carbon in that molecule, right? So this is 1, right? And then here again, you start on the right. 1, 2, 3, 4, right? Oh, I missed one. 1, 2, 3, 4, oh, 5 is here, and then 6 right there. Okay? So if we look at these things, it's just 5. Then there's the 4 and the 1. That's the beta 1,4 linkage that I was talking about. Okay? Beta 1,6 would mean it'd be the first carbon attached to the 6th carbon. Okay? And that gives them a lot of structural support, right? So if we think about glycogen, it's long-term storage of sugar for animals, and starch as long-term sugar for plants. Glycogen, there's a lot of branching. With starch, there's not as much, right? And so let's look at the way those things are formed real quickly, right? So I think they're very long chain carbohydrate. It can be hundreds of glucose in them, right? And they're put together because of the long chains of carbohydrates that are put together, right? Okay. So we talked about beta 1,4 linkages, right? And do I have my slide in here? I do. Okay, it's coming up. So if we think about this for just a minute, those carbohydrates then can add a lot of structural support to different organisms, right? And so does anybody know what this is? What is this? It's one of my favorite animals. What is that? It's a cicada. Very good. So, and well, actually the adult is still in it. I don't know if you can tell. It hasn't come out of the exoskeleton. It's still in it. So that is still soft. When the adult comes out of it, it's, it's going to come out of it. It's going to hang out for a couple of minutes. It's going to fly away. And it's going to make a lot of noise, right? These cicadas make a lot of noise in the evening. But the exoskeleton will stay behind. Those things are edible. They're just sugar. Mariel, what about it? If I bring one in, will you take a bite out of a cicada exoskeleton? I don't think they're annoying. I think they're beautiful. They're a sign of the end of summer, right? Because they're making those noise to attract a mate. They're going to no extra credit. They're going to mate. They're going to then go underground for a period of time, right? But Campbell said that chitin is also found in mushrooms, and that's true. And here, I put this here, of course, there are sutures now that are made out of chitin and other polymers that we can use, and then they just self-dissolve in the body, and you don't have to go and take out any of them. Yet they are tasty. I've eaten them a little bit baked, and I've eaten them cooked in a wok. They're good. Okay? Questions. When you see, this is one of the things that make me most happy in the world is when I see the pollinators doing their job, right? The pollinators are doing their jobs, and they're making end products, they're making honey. And honey, again, is sugar mostly. But honey has been used as a weapon against infection for years, right? And so you can put honey on wounds and things like that. And because of the tonicity of the honey's got a lot of sugar in it, what happens is the microorganisms that are growing will lose all their water. And if they lose their water, they die, right? Which is pretty amazing feat that this beautiful stuff that I like to eat on my pancakes or put in my coffee is very helpful in lots of different ways, right? So if we look at these things, right, you can see that they're arranged a little bit different, right? So here you can see with cellulose, right? The methyl group is alternating, so it's up, it's down, it's up, it's down, right? Good. It doesn't have any branching. The starch, you can see, has some branching, but the methyl group is up, 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 up. Are you with me? They're all on top. Okay? And with glycogen, you can see there's a lot of branching. That's the big thing. There's a lot of branching, right? And therefore, you can see that glycogen's a little bit different, right? Okay? Any questions? All right, and then we talked about cell-to-cell communication. I've already kind of made my little description about it when I talked about our cells and our tissues and our organs, and we talked about Steve donating his kidney to me, and how it wouldn't match up because of the fact that cell identity is going to be different, right? With the exception of the red blood cell, whatever you believe, right? If you believe in evolution, or if you believe in the big guy upstairs who created us, right? They did a good job, because when they put our blood system together, they either put no carbohydrates on them, or they put an A carbohydrate or a B carbohydrate, or they put an A and a B together, and that allowed us to be able to transfuse blood, as long as there was a good cross-type and match, right? I am not going to expect you to know, which types of blood can go to donor or whatever. I'm not going to know. I just want you to know that there's a whole science to really transfusing blood. It's not just, okay, give me some blood, right? To me, if I'm going to have an operation, as I did with my shoulder, I went and I donated my blood to myself, right? So I went in 2 weeks early. I donated a pint of blood, and in case they needed to transfuse blood, they would transfuse my own blood to me. And then after that period, if I didn't need the blood, they could go to where, because I usually donate a pint of blood about every quarter. And so that's one of the things I like to do now, right? So you can see how the carbohydrates are structurally put together, and the functionality of them, right? They mostly function in storage of long-term storage of carbohydrates. They could be used as fuel by the cells. They can be used as stability for plants and insects. And then they can also be used in cell-to-cell communication, right? Ladies and gentlemen, that's my talk on carbohydrates. We're going to talk about lipids next time. We'll start with lipids, and then we'll talk about proteins, and we'll end up with nucleic acids. Okay? So we're getting close to our first exam. Are there any questions? I will